Thank you, everybody, for coming to what is probably my last talk at Archbold Biological Station. I'm going to be retiring shortly in June, and I thought it'd be a good opportunity to kind of look back at the last 30-some years of the plant ecology program at Archbold and look at some of the patterns and progress, people, and publications. It's been an interesting year, but our program, these are the people in our program have done a great job carrying on with our long-term data collection with masks on in the field at 90 degrees and a lot of other things that we haven't had to do before. And about a third of the way through this period of time, this picture was taken with my friends and colleagues, Pedro Quintana-Ascencio and Carl Weekly. And just to emphasize that we're dealing with a lot of long-term data sets and quite a bit of aging process going on over the years. I'm going to talk about 6 themes in the plant ecology program. They're listed here. And I'll be talking about some of our main findings and most importantly, some of the people that have really been instrumental in research and conservation in these themes. And what I did for this talk is made a data set of our publications drawn from my own CV and from Archbold records. And the plant ecology program in this analysis is defined as publications by staff, collaborators, research associates, and research affiliates. They don't include visiting scientists that are not in one of these categories. I classify the articles into 1, 2, or 3 of the themes that I had up on the previous slide. I don't have a theme for conservation because it's probably part of 90% of the articles. I classified papers on demographic modeling separately from demographic papers that don't involve modeling. And I compiled some basic information that I'll show you in a few simple statistics and graphs that are shown here. And finally, when I talk about authors and whether they publish the most papers or not, I'm leaving myself out because it would be unfair for me to include myself since I've had such a great opportunity with the 32 years in this wonderful job at Archbold to produce science. Here's the graph that shows numbers of published papers in 5-year bins from 1990 to 2020. And this thing's gonna come up 6 times for each of the themes. So you don't have to worry too much about the details now. I'm gonna start by talking about vegetation science, which had peaks in articles in the early part of this 30-year period. And before 1990, most of the research at Archbold in plant ecology was in vegetation ecology and published by Ron Myers and Warren Abrahamson. And particularly Abe has been super productive. He's first author on 16 papers over this period of time. And his vegetation paper on Archbold is must reading for anybody that comes here, but he's had some really brilliant papers looking at fire responses of communities and plants, and then demographic patterns in palmettos. Palmettos are very interesting, being extremely long-lived, having high survival and low recruitment. And it's been an honor to follow Abe, and it's been a real pleasure to collaborate with him on several projects. So certainly a giant in vegetation ecology at Archbold. Early in this time, I worked with Nancy Kofeld, and we went shortly after fire and dug up a lot of plants and decided if they were coming back after fire by resprouting or spreading clonally or recruiting from seeds. And we found out that rosemary scrub had fewer resprouting species relative to scrubby flatwoods. That was one of the main conclusions. And rosemary scrub, of course, has many of the rare plant species that are found only on the Lake Wales Ridge. This paper was important because the data set that was in an appendix, which showed hundreds of species and how they respond to fire, has been used a lot to categorize species in designing experiments and analyzing other kinds of responses to fire. Christine Hawkes is quite involved with vegetation research at Archbold. And here's the oldest slide I'm going to show you of myself and Christine in 1993 when she was an intern here. And she's continued to work as intern, as a PhD student, and also then as a professor bringing back other scientists to Archbold, including postdocs, graduate students, and visiting scientists. And Christine and I published some of the early papers on gap ecology, including this 1998 paper, where we emphasize the important effects of fire and of microhabitats, the gaps among the shrubs, on plants of Florida scrub. Later on, Christine came back and did research on soil crust that was really basic and led to a lot of other interesting work over the years. Christine's now at North Carolina State University, and it's just been wonderful working with such a smart and fun person over the years. Christine and I and others came up with this conceptual model that you can see on the top here, where Florida rosemary, although it suppresses the growth of oaks and palmettos and herbaceous species, and we have a lot of experiments to show this, there's an indirect positive effect of Florida rosemary on herbaceous species through those two negative signs to the right and down back to the bottom here. And by controlling oaks and palmettos, Florida rosemary provides these microhabitats, these gaps that are very important for herbaceous species. This conceptual model also shows that Florida rosemary and herbaceous species have different requirements for fire, so that to coexist in the landscape requires diversity, variation in the fire regime, what we call pyrodiversity. So one of the mechanisms by which we think Florida rosemary is good for herbaceous species is through creation of gaps, because after a fire, the Florida rosemary plants are killed and come back very slowly, and the open areas that are devoid of resprouting palmettos and oaks are great microhabitats for smaller plants. And we've worked on these gaps in a whole number of papers over the years. Most recently, in 2017, we looked at dynamics of gaps over 8 years. And we found that gaps that are burned became larger and merged, and gaps that are unburned became smaller and fragmented. This is a pattern that we knew existed, but it was nice to see it in the same areas over time. And this is important to communities of plants and to populations of plants, so that species richness on the y-axis here increases with the area of the gap, but the increase is faster in areas that burn during this 8-year period of time. That's the solid line with the steeper slope, and that's the significant interaction term in this model here. So fire is important in promoting species richness, the number of species in these gaps, and in individual species. So this pinweed here, Leccia decurtii, generally only went extinct in gaps were unburned, and colonized gaps generally if they were burned. And this is despite the fact that this plant, individual plants are killed by fire, but it comes back from a persistent seed bank, and the population dynamics of the species are benefited by fire. The second theme is population ecology, and that's the white circles here, and accounts for the most of the publications, or more of the publications, in any of these other themes. This includes pollination ecology and studies of genetics. And I started in 1988 doing demographic studies on Dicerandra frutescens and Eryngium cuneifolium. They grow in different kinds of scrub habitat, but they're similar in having preference for gaps, a positive response to fire, and a long-lived persistent seed bank in the soil. And over the years, we end up studying about 18 species of plants doing intense demography. This graph here shows 14 of them are annual survival on the y-axis. Species like scrub plum on the upper left have high survival, and it's not very variable. And the dots here represent different years over time. Pelagula lutonii, this beautiful little herb here on the bottom left, has very lower survival and a lot more variable. And this kind of data across many species, but with intensive work over a long period of time, is rather unusual, and is, I hope, a jumping off point for continued analyses by other scientists at Archbold. Now, to collect this data requires really hard work, and I want to thank all the research assistants that have helped, including all the folks that are listed here. Lexi and Scott are my current research assistants, and then going way, way back to Alea, and other research assistants that I didn't have good pictures of, like Becky Yar and Margaret Evans. And I'm also going to call out three specific research assistants, Carl and Stephanie and Stacy, later in the talk. But to collect this data year after year, to train interns to help collect the data, requires a lot of persistence, a lot of attention to detail, a lot of organizational skills, and these folks all help with that. In particular, Carl Weekly, who is my longest running research assistant, here with me for, I believe, 15 years or more, is been the first author on 12 papers, which is the third highest, and has been an author on 21 papers since 1990. And I met Carl long before he worked here, and we started a collaborative project at a Lake Wales Ridge State Forest, and they put up a big billboard about that project, and so we're posing in front of that. But Carl, as those of you who know him know, he's really smart, really determined, and super organized, and just couldn't have done very much of this without him. So thank you, Carl, and hope you continue to enjoy your well-deserved retirement. Now, we began to do some ecological modeling once we got a decade or so of demographic data, and the red triangles here show the number of papers we've published on ecological modeling over the years, and it's been a kind of consistent paper or two a year. And the gentleman who's most responsible for this is Pedro Quintana-Ascencio. I met Pedro in Mexico at a conference, and we bonded over chili rellenos, I believe, at a Mexican restaurant. And Pedro came and did his PhD here, later on returned as a postdoc, later on as a visiting scientist from the University of Central Florida. And we had a steady stream of interns and research assistants back and forth between our programs. Pedro is the first author on 10 papers over this time, second most of anybody, and an author on 34 papers, which is the most of anybody. And if you know Pedro, he's super enthusiastic. He loves the field, but he's really also a talented statistician and modeler and just a great collaborator. So, and Pedro's still active and will remain active in many Archbold projects for the next number of years. Now, Pedro and I and our collaborators use a variety of approaches in demographic modeling, starting with matrix models, going up to integral projection models, metapopulation models, and really focusing on drivers of demography. We have demographic models of about 10 species, including Chrysopsis highlandsensis, our latest one, which Stephanie is working on, and Laetris olingeri, which one of Pedro's students, Matt Ty, did a lot of work on. And these data sets have been shared also with other folks to do different kinds of demographic modeling. So this is one of the latest examples. Most of these authors are from the United Kingdom, and they used stochastic integral projection models with factor analytics using our data, and find, for example, that the population growth rate on the y-axis for Eryngium cuneifolium is highest the first decade after fire and goes below zero, so with predicting declining populations by 15 years after fire. This is consistent with earlier work we did, but using more data and a more sophisticated modeling approach. This again suggests that this Eryngium, which coexists with Florida rosemary, does best with frequent fire, but it's in rosemary scrub, which burns infrequently. So it's not going to be found in every patch, and it's going to be moving around the landscape over time. More recently, we've scaled up our demographic modeling to landscapes, for example, with Hypericum cumulicula in a series of papers, one of which was just published in 2019, adding landscape variables like the position of rosemary scrub patches in the landscape and then relative elevation. We work with Viz Slater in creating layers of elevation, and in particular looking at the relative elevation from the nearest wetland boundary on the upper left to higher elevations in rosemary scrub. You see on the right, there's about a 4-meter elevation gradient here. And it turns out the elevation of the plots that support Hypericum affect the vital rates and the demography of this plant. So we do modeling that Pedro and others did, showed that shortly after fire, population growth was greater at higher elevations, that's this black line, and then by 15 or 20 years after fire, those are less favorable sites for population growth. So there's an interesting interaction here between the microhabitat and the fire affecting demography of this plant. Well, we've been talking about fire, and it's the dominant ecological disturbance in Florida scrub, and it's also been a very important part of our research. The green triangles show we've had consistent output of around 10 papers every 5 years that dealt with fire ecology. And I want to thank Kevin Main and everybody else that's helped with prescribed burns, because without aggressive fire management and without great cooperation between Kevin and the researchers here, we would not be able to do the kind of fire ecology research we're able to do at Archbold. This goes for plants, but also for gopher tortoises and scrub jays and other species that have relationships with fire. So thank you, Kevin. And I mentioned already Eryngium in relation to fire return intervals. And for about 8 species, we're able to identify optimal fire return intervals by running models with fire as a driver at different frequencies. And this shows some of the papers over the years that have been produced using that approach. And you'll notice that these different species on the slide here have different optimal fire return intervals, and that also an argument for pyrodiversity in fire management. Dicerandra frutescens on the lower right has been a subject of at least 3, maybe 4 papers looking at optimal fire return intervals using different modeling techniques. And this is a plant that's protected largely at Archbold Biological Station and does require fairly frequent fire. So it's really important to get this right to keep this plant from extinction. We've also worked with fire at the vegetation or community level, asking whether fires can be too frequent or too intense for resprouting shrubs like oaks and palmettos that are the foundation species of scrub. And if you walk through recently burned scrub like Reed did before he took this beautiful photograph, you'll be impressed at the ability of species like saw palmetto to resprout after fire. So we did an experiment that took 10 years. We had 6 years of fire treatments and 4 years of following up, looking at fires that occurred every year, 2 years, 3 years, or 6 years, and the biomass response of about 8 species of shrubs. And we did find, if you look on the left, a more muted recovery. When you look at the recovery after fire, the black is pre-fire. Look at the recovery. So frequent fire does suppress the recovery of shrubs. However, they're still recovering. They're going to get back to where they were. So our main conclusion from this is that really frequent disturbance are not going to have a big effect on the resprouting plants. They may have effects on plants that don't resprout. We also did a long-term study over a decade with dozens of fires looking at fire intensity effects on resprouting. We use these hobo data loggers you see in the picture to measure temperature at 2-second intervals, calculating 1-minute means and residence times. This paper is going to be published soon, I hope, in Fire Ecology. So we find that Florida scrub has a lot of heterogeneity in fire intensity and also very intense fires. You see maximum 1-minute means commonly between 200 and 600 degrees C. This is a lot hotter than fires in other ecosystems. And you'll also see residence times of often 10 or 15 minutes. So there's a lot of heat being applied to these plants. Their meristems, though, are in the sand, and the heat doesn't seem to affect their survival or growth. So the graph here shows percent survival as a function of maximum temperature, and even small resprouting plants, herbs or shrubs, seem to survive very well. And it doesn't seem to affect the growth very much. There are differences among species. So saw palmettos recover their biomass a lot quicker than oaks. And anybody who's walked around a scrub 6 months or a year after fire will notice this. And this graph here shows the height recovery first 7 years after fire. And the other thing that this would show is a slower recovery compared to coastal scrub that Paul Schmalzer has done a lot of study in. And our soils are less fertile, which may account for the slower recovery of the resprouting shrubs. The second to last theme I'm just going to touch on briefly is combining physiology and ecosystem ecology, which are related in plants. And I'm just going to point out we have a number of different aspects of this that we studied. Early on, with Mark Abrams and Noreen Gallo, we worked on water relations of plants, finding that fog relieved water stress in scrub plants. Sonali Saha, my postdoc, and others, we were able to look at rooting depths and document 3-to-1 ratios below ground to above ground rooting depths in these shrubs, which other visiting scientists have done similar things. So a lot of the action in these plants is below ground. Carl Weekly and others looked at soil water dynamics over a long period of time. Soil water, of course, responds to precipitation, but gaps and recently burned areas, the soils tend to have higher moisture than areas with a lot of shrubs, suggesting transpiration loss is higher than evaporation in our ecosystem. Jenny Schaefer, I'll mention later, has done a lot of work on nutrient dynamics in relation to fire. And finally, really exciting research recently by our collaborators at the University of Miami. I saw Chris Searcy on this call. Great work on microbes in Florida scrub and how they affect the growth of some of the plants we've been studying. The final topic I want to talk about is restoration and translocations. I'll mostly just talk about translocations. And that's the graph here with the blue squares, and we haven't published very much on this yet, but I think we've done some good conservation work. I want to call out, besides Carl Weekly, Stacy Smith, who's an outstanding research assistant with me for more than a decade and engineered these translocations, which are logistically really challenging. So Stacy, it's always a delight to have around and did some really, really hard work for us for a long time. And also Stephanie Koontz, who you've seen already, a current RA who's also been involved with these augmentations. And here, Stephanie and I are celebrating getting some grant funding, which allows us to continue this work. And I'm going to thank some of the funding agencies at the end of the talk. So thank you, Stephanie. Over the years, we've introduced 5 species to different sites. I'm going to mention briefly 2 of them, the ones in yellow. Florida Ziziphus, a really imperiled plant that rarely reproduces in wild populations. We've done 12 translocations over several decades. And the translocations are intended to mix different genotypes that can produce fruit. The material comes from Bach Tower Gardens and our colleagues there, including Cheryl Peterson. And we've been following the demography of these translocations. And you can see from the pictures here, a lot of people are involved. It's a lot of work to grow the plants and put them out. What we found is that we've got similar survival in our introductions and augmentations, but they're lagging behind in growth and reproduction. And it's going to take many, many decades for us to really determine whether these Ziziphus translocations are successful and can create new viable populations. But nonetheless, the introductions are helpful in guiding us to improve our translocations over time. More gratifying, the short run is short-lived plants like the mint Dicerandra chrismanii, where we've done introductions and augmentations at properties owned by U.S. Fish and Wildlife Service. And this graph just shows number of seedlings recruited over time. These solid lines on the left are wild populations, and these lines up here are introductions and augmentations. They've just gone crazy and produced second generation and beyond. And so it looks like Dicerandra chrismanii and the other Dicerandras are pretty tractable in terms of augmenting populations or creating new populations. I'd like to thank all the young people that have been such a help. There's pictures here of very successful young folks like Katie and like Oru, the left and right. In the top middle, a really remarkable group of interns that all presented at a conference, and some other folks here. And my current interns, Haley and Aaron, who've just done a great job under difficult circumstances this year. And here they are finding new seedlings of Dicerandra thunicola. Our interns have produced 49 papers, which is a quarter of our papers have been intern authored. And I want to thank Mark Dayrup, who has helped mentor many of these interns with projects with pollination ecology, where we certainly need his help. And But most of all, thanking the interns for all their new ideas and all their youthful energy and the fun that they've created. It's been great to have them. And a couple intern projects, just briefly, Jamie Peeler looking at serotiny and sandpine, finding serotiny decreasing with tree age, a really interesting result, which may be due to bed hedging for variable fire regimes, but it could be due to the high maintenance costs of maintaining living pine cones on the tree. And Rachel King with a great project experimenting with 6 species with different levels of smoke and heat. The graph here shows Eryngium cuneifolium with some smoke stimulated germination in the absence of heat. And finally, a whole bunch of graduate students. I've mentioned some of them already, but Jenny Schaefer on the left, Mary Carrington on the right, each have been first author on 7 papers since 1990, starting with their grad student projects and continuing on with follow-up projects over the years. And they're both still active with research at Archbold. We've had great relationships with agencies and nonprofits. And if you know all the acronyms, I'll buy you a beer sometime. But the people that have been involved, including, I'm not going to name them all, but I'll just call out Matt Vance at FWC and Dave Bender and Todd Mecklenburg at US Fish, and all the great folks at TNC, including B. Pace Aldana, fantastic people at Forestry that have worked with us, the EPAC folks, NSF, Cheryl Peterson at Bach, Suzanne Kennedy, a great collaborator, and there's many, many, many more. And I certainly hope that Archbold and the Plant Ecology program will continue these great interactions. To summarize conservation lessons from our research most broadly, I'll say that we believe that long-term intensive research is important to capture scrub plant population dynamics. These results can be scaled at the landscape level using models, and that introductions and augmentations can help recover species. And the other real threat is the importance of fire to scrub plants and to vegetation structure that promote biodiversity, including the effects of fire creating and maintaining gaps, and the resilience of resprouting shrubs to frequent or intense fires, and the need for biodiversity and burning to allow the coexistence of species with different life history responses. Well, this whole talk's been about acknowledgements, but I'd like to acknowledge my family. And I'd like to acknowledge all the collaborators again, all the RAs, interns, and more shout outs to Archbold. I'd like to acknowledge Roberta and Viv for their work with GIS. Hillary Swain, a great director, and I just really enjoyed reporting to Hillary over the years. She's challenged me and made my work better. Again, Mark, the other program directors, Reed, and the 2 Betsy's. I'm very proud of Betsy Bowden, who used to be an intern and now runs the science program at Buck Island Ranch. And finally, all the great plants and animals and the Florida scrub. It's been an inspiration. And that's all. Thank you.
Thank you very much, Eric. That was wonderful. I'm looking in the Q&A. Does anybody have any questions? I know that's a huge overview from his 30-year career here, and there's a lot of things that we've learned, but also many questions that could come out of that. We only have about a minute before we're going to move on to the next person. There is a question here from Amy Favier. She says, amazing work as always. I was curious about the intensity and length of the fire that you measured in the soils with the hobos. You mentioned those are intense relative to location. Can you also address that?
Hi, Amy. Thanks for the question. The fire intensity we measure was at the soil surface, and we have measured fire temperatures in the soil. Mary Carrington's done quite a bit of that, and the heat really attenuates in the soil. So that's one reason that these resprouting plants can survive. I think the other part of your question might be the spatial variation in fire intensity, and it's really great. So you can get much different fire temperatures and residence times just a few meters apart, and that's something that's important to take into account in looking at fire responses. And the variation in fire intensity across space in Florida scrub is probably also important to maintaining the diversity of species that we have.